Imagine having the power to rewrite the fundamental blueprint of human life, not just for an individual, but potentially for generations to come. This incredible capability, now within reach through technologies like CRISPR gene editing, forces us to confront some of the most profound ethical questions humanity has ever faced. Embryo genetic modification raises a complex array of ethical issues primarily centered around safety, the moral status of the embryo, potential societal inequities, and the very definition of what it means to be human. The core mechanism behind these concerns lies in the nature of germline editing. Unlike somatic cell editing, which only affects the individual treated, germline modifications are made to reproductive cells or early embryos. This means any changes become heritable, passed down to future generations. This raises serious questions about our right to make irreversible alterations to the human gene pool, especially when the long-term safety and potential unintended consequences, like off-target edits or mosaicism, are not yet fully understood. Furthermore, the varying views on when an embryo gains moral status directly impact the permissibility of such interventions, with some arguing for absolute protection from conception, while others emphasize the potential to prevent debilitating diseases. Navigating these complex waters requires a multifaceted approach. Ethicists, scientists, and policymakers are working to establish robust regulatory frameworks and international guidelines. This includes careful consideration of the distinction between therapeutic uses, aiming to cure or prevent serious genetic diseases, and enhancement uses, which might seek to confer non-medical advantages. Establishing transparent and inclusive public dialogues is also crucial to build societal consensus and ensure that any application of this powerful technology reflects broad ethical values. An important distinction often discussed is between germline editing, which alters the genes in reproductive cells and is passed to offspring, and somatic cell editing, which targets non-reproductive cells and only affects the treated individual. Understanding this difference is key to appreciating the magnified ethical stakes of embryo modification. Another concept is the principle of proportionality, which asks us to weigh the potential benefits of an intervention against its risks, especially when those risks involve future, unconsenting individuals. The fear of a slippery slope, from preventing severe disease to creating designer babies, also looms large, challenging our understanding of genetic determinism. Ultimately, the ethical issues surrounding embryo genetic modification demand a cautious, thoughtful, and globally coordinated response. By grappling with questions of safety, justice, and the very essence of human identity, we can strive to harness this revolutionary technology for the profound good of alleviating suffering, while simultaneously safeguarding against unintended harms and ensuring a future that is equitable and respects the inherent dignity of all.